Hello everyone, and welcome to today's live broadcast, Planet-Scale Analysis with Millions of Genomes, presented by Dr. Alexander Waite Zaranek. I'm Dr. Michael Chow of Harvard Medical School, where I am the Director of Human Subjects Research for the Personal Genome Project, and I'll be your moderator for today's event. This educational web seminar is presented by LabRoots. You can learn more about LabRoots by visiting labroots.com. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you like at any time during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Also note, this is an educational webinar and offers free continuing education credits, PACE, RACE, CME, or CEU. If you'd like to obtain credit after the webinar is over, you may click on the CE button located on the bottom left corner of your screen that will lead you through the process. And now it's my pleasure to introduce Dr. Alexander Waite Zaranek, who I've known since grad school and who I've worked with for many years now. Dr. Zaranek is co-founder and chief scientist at Curaverse, a venture-backed company focused on building a free and open-source platform for storing, analyzing, and sharing biomedical data. Alexander, or as I've always known him, Sasha, works on open technologies that are part of the revolution that reduced the human DNA sequencing cost by 1 millionfold since the completion of the Human Genome Project. He's been a leader and strong advocate for open software and genomic information throughout his professional career. His contributions have led to highly cited publications in Science, Nature, The Lancet, and other leading scientific journals. Recently, he presented his work at the White House Open Science Champions of Change event. Currently, Sasha's research includes the development of clinical quality applications for processing massive data sets spanning millions of individuals across many collaborating organizations. Together, these data sets are expected to reach exabytes in size. Prior to co-founding Curaverse, Sasha was a co-founder and director of informatics at the Harvard Personal Genome Project, where he continues to serve in that capacity. Please join me now in welcoming Sasha.
Thanks so much, Mike, and thanks again to LabRoots. So today I will tell you about planet-scale analysis with millions of genomes. And we're going to start with a very small piece of one genome, my genome, actually. And if you look at the little circle, there's a C there that is different in my genome from most people's that are watching the seminar. This particular change is in the APOA5 gene, and it leads to abnormal triglyceride levels. I have them, my mother has them, my grandmother had them, so it's a very real thing in our family. But interestingly enough, this change is not associated with heart disease, even though high triglycerides normally are. Genomes are complicated, and understanding them is going to take a huge amount of work. This is just one feature in my genome, and there are millions of features like this. So we're going to have to look at slides like this for a very long time, probably several years, if we want to look at my genome. And if we want to look at millions of genomes, then you can do the math. So we need to do something different from that. Luckily, you don't have to worry about memorizing my genome or listening to me talk about it. You can just download it from this URL. It's actually publicly available. I have whole genomes from saliva, from blood, and exome, all with different platforms available at that URL, as well as phenotypes and many other things. So you may wonder, why would I even make my genome public? But the reason is that many, many people are going to have to share their data if we're going to understand what genomes mean. And most people's data will be private, but it's incredibly helpful to have some genomes that are publicly available so that everyone can have a starting point that is shared by the whole community. So in the next 5 years or so, we're expecting about 5 million whole genomes and many more genotypes and exomes. This is going to be 500,000 terabytes of raw data, so about an exabyte. And that's an awful lot of data. How are we going to handle all of this? Well, what I like to do is I like to build on the commons. And this is something that has been happening for decades now in the internet scale businesses. Linux powers your Android phone. The BSD daemon powers your iOS phone. Virtually all internet scale businesses use free software to power their planet scale data centers. And so a big part of the commons is free and open source software. And it's something that academics in life sciences are only really getting used to using now. And pharmaceutical companies as well are really just starting to get used to the idea that the free software is something that they should be using to build on for their work. The other important part of the commons is how my genome was able to be shared with you. So as Mike already mentioned, we're co-founders of the Personal Genome Project, George Church's project at Harvard Medical School. And these are the first 10 participants in the Personal Genome Project, which when we started in 2005 was still an incredibly audacious idea. At the time, we wanted to make this data available to the participants themselves, and of course to the general public, not just to the research elite. There's an entrance exam to make sure that people really know what they're doing when they make their data public. And the hope was that we would scale the project when we reached the target of about $1,000 per person for DNA and trait data. And we've actually reached that target this year. So there's a company called AirVatos that is now offering $1,000 genomes to any consumer that wants to get their whole genome sequenced. And this is something that's, of course, available to PGP participants. In addition to making genomes and phenotypes available, we also wanted to enable highly integrated holistic systems biology. So if you needed cells from participants, we hope to make them available. And today, there are now hundreds of cell lines available from PGP participants that you can use in your own functional studies and experiments. So building on the commons, software and data, public data, we can ask this question: how do we organize computational resources to serve the needs of scientists, physicians, and the general public? And I've been thinking about this question, for about a decade. And one of the things that is so challenging is that when you have exabytes of data, you can't necessarily make copies of all of it. So if scientists wanted copies of the exabytes and physicians want copies of the exabytes and the general public have their own needs that need copies of the exabytes, it really just gets way too expensive. And so we need to think about ways that we can create computational infrastructure that could answer these different constituents' needs at the same time. But where are we today? Well, if you work in a lab and you work with genomes, you've probably seen piles of hard disks sitting on lab benches. These are old hard disks, and so they're a little bigger than the hard disks you might get today. But hard disks are hard disks. They're terrible to work with. They break, they overheat. It's nice that they can be mailed to you, but imagine that you had to work with millions of these hard disks in order to do your analysis. It's completely and utterly impractical. We need something better. We need a platform, something that can meet the needs of of all of the different constituents that I mentioned. So we've all heard about big data. We know that internet scale companies have been working with big data for a very long time now. But biomed does have unique requirements. There are sharing requirements, there are governance requirements, and there are provenance and reproducibility requirements, and unique data requirements that make biomed data special, that will mean that our platform has features that may not be available in other internet scale big data platforms. So the first of these is sharing. Biomedical organizations inherently need to share. Today, when a doctor sees a case that they don't know how to solve, they'll call their colleagues, sometimes on the other side of the world, and they'll say, hey, do you have a patient kind of like mine? This has to happen at this scale where now we're starting to generate millions of genomes. And you just can't pick up the phone and ask a question about 1 million patients. So sharing is a challenge. The way we think about sharing is to enable federation. We'd like data to stay where it is in the country or in control of the organization that generated the data. And then we'd like questions to move to the data. So applications can move through a federation, and the question can be answered in the control of the organization that generated the data. Federation, of course, could be optional, or it could be used within an organization. If you're a multinational company, you have sequencing and health data all over the country, or all over the planet. You simply can't move the data out of the jurisdiction because of local regulatory requirements. So you need federation even in a single organization. And of course, data from the Personal Genome Project and other public data can be a resource that everyone uses as well. That there's another need for sharing. Now, Larry Page and Mark Zuckerberg, who both have amazing planet-scale computational infrastructure, that, certainly the most sophisticated in the world, that planet-scale infrastructure doesn't need to be used for sharing. Mark and Larry don't have to share their customer data with each other. Biomed is different. We could spend this entire talk on governance. Security and governance is extremely important in healthcare. Health data is mostly private. It's not going to be the case that people will generally make their genomes available, with notable exceptions. And so the governance and security requirements around this data are enormous. And so they really need special attention, jurisdiction by jurisdiction, organization by organization. Provenance is also very important in biomed. So this is Charles Darwin. All life on Earth is connected through evolutionary processes. And you can think of those processes as having a kind of provenance. You can see how things are connected to their common ancestors. And we need this in biomed. But again, internet scale companies don't need to worry about this. So when Google and Facebook are handling queries from their customers, people are searching for Paris Hilton, and they're searching for cancer, and they're searching for medicine. And these queries need to be answered very quickly. And if they can't immediately answer the exact provenance of why a particular search result was presented to a particular person 10 years ago, this really doesn't matter for the internet scale businesses. But in biomed, if a life or death decision was made for a patient based on a report that was generated 5 years ago, then exactly how that report was generated is important. The provenance of that report is important. And so biomed is different in this way. The data in biomed is also unique. You know, genomic data is very interesting because we understand it on an atomic level. We could write down a genome almost atom by atom, but we certainly wouldn't want to do that. That description would be incredibly verbose and in some sense completely useless. And so we want compact descriptions that take advantage of the uniqueness of biomedical data. When I think about weaving these requirements together, we get the AirVatos platform. This is an open source platform for managing and analyzing biomedical big data. It's been running at the Harvard Personal Genome Project since 2007. This is kind of the simple schematic architecture. So if you start at the top here, you have applications that run the Personal Genome projects. You have applications that support precision medicine, patient care, diagnostics, pharmaceutical research. And all of these applications, no matter where they're running, no matter where on the planet they're running, have common features accessible to them: data management, analysis, provenance, governance, sharing. And then underneath this, you can run AirVatos anywhere. It can be, because it's open source, it can really be made to run ubiquitously. It can run on Amazon Web Services and does today. It can run on Google Cloud Platform, on Microsoft Azure, on OpenStack, and all sorts of different on-premise configurations. So it can run privately in your own data center. It can run in the public cloud. And so the idea is that we want a platform that will run wherever the data is. And that's why it has to be open source, so it can be modified to meet the requirements of the local configurations. This is a technical diagram. The details are not important. Of course, a system like AirVatos is quite complex. But the important thing here is the URL. If you visit airvatos.org, you'll find, and the GitHub page, you'll find all of the code available. You'll find all of the plans of how the AirVatos project is evolving, what's going to happen in the next 2 weeks, what's going to happen over the next few months. There's active contributors participating from all over the world, from Russia, from the UK, from various parts of Asia, and so obviously all over the United States and Canada. So you can absolutely just go to this URL and start participating in the community. All of the software is free, no cost. You can just download it. Now, the way that AirVatos gets used should be pretty familiar to people who are used to any kind of sort of data-intensive science. So in the upper left here, we have images. This is called primary analysis that's coming off a DNA sequencer. These are converted into ACs, Gs, and Ts. These are called reads. That's secondary analysis. Then secondary analysis leads to tertiary analysis, where you have questions about specific genetic features, like the one that I showed you in my from my genome at the very beginning. So Lightning is this component of AirVatos that lets you query all patients with similar phenotypes, genotypes, and outcomes at planet scale. It needs to incorporate the spectrum of human genetics nomenclature. People have been using genetic information to diagnose patients and to treat patients for decades. And doctors, when they called up their colleagues, would use these nomenclatures, ISCN, HGVS, Hugo, and so on, to talk to each other about genetic features in their patients' data. We want to be able to support all of these nomenclatures in Lightning, and so we're working with a community to do that. For those people who are not familiar with these kinds of diagrams, these are cytogenetic maps. And they're sort of stylized pictures of what DNA looks like when you stain it and look at it under a microscope. People who are only familiar with next-generation sequencing think of genomes as being little ACs, Gs, and Ts. But obviously, people who worked with genomes several decades ago thought of them more like this, as karyotypes. So how do we support all of this nomenclature in Lightning? So we use a concept called tiling, which segments the genome and provides a common and consistent coordinate system. Tiling begins by choosing a tag set. It's a group of k-mers. We make them 24-mers. There are at least 2 bases from each other, and they're each found exactly once in the pangenome of the species. The tiling that we use today has about 10 million tags. So given one of these encodings, we can convert a genome into tiles. And I'll show you how this works in a little bit more detail. But if you remember the tile that or the feature in my genome that I originally showed you, you can think of that. You can think of that as a tile. And if a change falls on one of the tags, then you skip ahead to another tag. So here's the little bit of my genome that I originally showed you, with the C that leads to high triglycerides in my family. Now I've added tags, and then I've reorganized the bases into separate tiles. So there are 7 tiles in that original slide, and the one in the middle is the one with the C. Now I'll just focus on that. This is a single tile, the one that leads to high triglycerides in my family, and the sequence of letters and numbers that's underlined at the bottom of my slide is a consistent name. It's actually a cryptographic hash that's generated from the ACs, Gs, and Ts in the tile. And this is a unique name for this particular feature in my genome. And so using this technique, you can build up a set of unique tiles for the planet's genomes. And every lab that uses the same tag set will always get exactly the same names for the same sequences. And this allows us to, in a very scalable way, a way that's scalable to millions of genomes, unambiguously talk about genetic features with our colleagues without having to worry that we have misinterpreted what they were saying or misapplied the sort of more doctor-friendly, more human-friendly nomenclature. And we can layer the human-friendly nomenclature on top of these IDs. There are 3 views in this slide. The top view is sort of a standard genome browser. The one in the middle is actually a graph that allows you to visualize a population of tiles, just for a very small portion of a genome. And then the last part of this, at the bottom of the slide, is a single tile. This particular tile is an extremely serious pathogenic BRCA2 mutation, and this is found in a PGP participant, and this person reported having breast cancer to the project. And there are many details on tiling and so on at the preprint at the bottom of the URL. Once you have genomes encoded as tiles, you can also do machine learning. So this is extremely easily because genomes become arrays that you can work with very, very easily. So here you have a simple principal components analysis. The x-axis is the first principal component, the y-axis is the second. And if you take 680 whole genomes from public data and you just plot them this way, they just separate quite naturally. And when you color code them, it turns out they cluster by ethnicities. So you can, this is a very simple example, but you can also use this data to do many, many other things. So one of the data scientists that works with me, she got really quite enthralled with this encoding when we discovered that from just a handful of Personal Genome Project genomes, she could identify the specific part of the genome that was responsible for blood type. So that's a very simple machine learning exercise that in the past would take a very, very long time, many sort of PhDs worth to actually isolate the gene that was responsible for blood type. So once again, Lightning queries are federated. So the idea here is that everywhere that you have AirVatos instances, you also have Lightning instances. And you can ask questions of each other's genomes. You can say, do you have this tile in your genome? Or can you give me all of the genomes that have this set of phenotypes? Or maybe this combination of genotypes. And every organization that receives those questions can decide for themselves, are they authorized to answer the question or not? The data never has to leave the control of the organization that sets up the AirVatos instance and the Lightning instance. So as I've already said, AirVatos, the Personal Genome Project data, all of these things are open. We have been developing open standards alongside these software and data projects. So we have quite a lot of participation in the Global Alliance for Genomics and Health, which is a standards organization that is trying to emulate the W3C, which is developing standards for the internet. I urge you to take a look at the GF4GH. Also, all of the questions that I've been talking about that AirVatos and Lightning allows you to ask and answer, these are described in a standard called the Common Workflow Language. This is a very popular sort of grassroots effort by many organizations and bioinformaticians, now several hundred bioinformaticians, that is allowing bioinformatics pipelines to be portably shared across organizations. So these sorts of standards make it possible to present Personal Genome Project data, to present software like the AirVatos project software in a way that can be used even more broadly, by people who want to just interface with these things, but not necessarily use AirVatos themselves or not use the PGP data, but by using standards like from GF4GH or the Common Workflow Language, these things are more broadly accessible by taking advantage of these standards. So I think that's it for my presentation today. I hope to answer your questions. Thank you so much.
That's great. So now we're going to move on to the Q&A section. I want to remind you that it's time, if you have a question and you'd like to ask, please do so now by just clicking in the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. And so let's get started with our first couple of questions. As our questions are coming in, I have a question for you, Sasha. First of all, that was a great talk. if somebody in our audience wanted to look at your genome and sort of explore it, or any of the genomes, could they find, for instance, that ApoE5, ApoE5 mutation? Could they find that by looking at that URL that you gave?
Yes,
Yeah.
yes. So you can actually download copies of my raw data, and you can also get copies of genome reports that interpret the data. So you can do that for my genome and for hundreds of other people's genomes. So there's really a growing, interesting corpus of data that is completely public and unencumbered. And so we've had actually a lot of interest from high schools that want to work with this data, and they would never be able to work with this data if PGP project participants were not willing to share.
To share, yeah. And how much is an exabyte?
So an exabyte's a million terabytes, and a terabyte's about one of those hard drives.
Okay.
So
great. So one of our first question is, thanks for the great talk on planet scale analysis. I have a question about where you think we are with the standardization of tertiary analysis, such as alignment and variant calling tools. Is there a set standard? If not, how and when will we get there? Thank you.
So I think that this is a really important moment with projects like the Genome in a Bottle initiative that's run by the NIST, the National Institute for Standards and Technologies, to really standardize what a genome is. Because I think it's really quite surprising to the general public that we've never actually completed every single base in even a single genome. And so we have a lot ahead of us to actually get to a place where we have reference materials that you can just order from NIST and sequence yourself and see, does my analysis lead to the correct answer? So NIST has shared about 70% of the genome, which they call confidently called. But until that other 30% is finished, we're not going to have standardized pipelines. And so these standardized reference materials are essential to make that happen.
Mm-hmm. Okay. Another question is, where could I find open source data for mapped genomes? I'm not sure. Maybe that question is a little bit redundant with mine. Where could I find open source mapped genomes?
Well, the URL that I mentioned at the beginning of the talk does have everything from raw data, raw reads, all the way to interpreted genomes. So you can see map genomes from the Personal Genome Project.
Okay. Next question is, happy to see the open source platform for genomics research. Unfortunately, most of the databases and software are getting commercialized. What is your comment on ethical perspective of making all of them open source?
So, I work at a venture-backed business, and as well as being a co-founder of the Personal Genome Project. And I think that what people are not used to in life sciences that has been long established in information technology is that infrastructure can be open source, and then proprietary innovation can occur on top of that infrastructure. And so there is no reason why open technologies can, in a very sort of vibrant ecosystem, coexist with proprietary innovation. And that's actually separate from the fact that even if your software is open source, you can still have a very successful commercial business around that. So most IT companies these days that are making new storage systems or making new other kinds of new machine learning technologies. These are based on open source software, which you can actually download at no cost, but these are venture-backed businesses. So in life sciences, this is still a new concept.
Yeah. Okay. Let's see. Another question is there a software tool repository? My research group can make contributions to it. Is that possible? What are the requisites to be part of this initiative?
So everything is happening completely in an open source distributed way. You can go to GitHub and participate there. You can go to AirVatos.org and participate there. There are no requirements on you at all. And the way that if you've never done open source development before, it tends to be a kind of ad hoc meritocracy where what you do is you take the code on GitHub, you get it running, it doesn't do something you want it to do, you add a feature, you talk about the feature in the forums, other developers comment on your implementation, and then eventually you do something called a pull request where you say, hey, I've really gotten this working, can I contribute this code back to the project? And sometimes there's additional conversations before that pull request is incorporated. But that's kind of how the open source development works.
Yeah, I think maybe the question was from the point of view. Suppose you have some sort of software that your lab, yeah, something you've developed completely, and you want to add it to the AirVatos platform or
So
repository.
I think that we are still working on how that will work. So we have collaborations with the Galaxy project, with the AMP lab that creates Spark and much of the sort of distributed computing infrastructure that powers the internet. Each of these collaborations tends to be a little bit different. And so the best thing to do is to just go either email us or visit AirVatos.org and just say, hey, we have some tools we'd like to contribute. And we'll find the right place for them.
Okay, sounds good. I'm not sure I understand this next question clearly. It says, I would like to know how we verify the genome in different methods from human beings. Perhaps the question is about other species, or is there any challenges that verify the different methods from human beings? So maybe other species. Does your platform support that? Does Lightning work with other species, for instance?
So for the time being, we are very focused on humans. But there's no reason that you couldn't tile non-human genomes. And actually, we have interest from contributors that want to develop tilings of non-human genomes. So one of the things that open source projects can do is connect people who are interested in contributing with one another.
I wonder if you could use ancestral data, like GIMP or other primate data integrated with your tiling and find out, not only what the principal component analysis, but also ancestral alleles versus more modern alleles.
Yeah, there's a project called the Monarch Initiative that looks across all life. And so we hope to integrate with Monarch over the next few years.
Cool.
So.
Another question, is AirVatos mainly for scientists, bioinformaticians, and IT at institutions? Can an individual with interest in environmental factors of genomic research use AirVatos?
So
Let me go back to the slides.
Back in this earlier picture,
1 second.
I mentioned that AirVatos is a stack that allows precision medicine applications to run ubiquitously wherever the data is generated. The Personal Genome Project has two applications that are in the top right corner here, Tapestry and Get Evidence, that run the project. And our hope is that ordinary people who are interested in genomics will be able to use these applications, not just PGP participants, but any member of the general public. And, you know, the reality is that all projects like this are limited in resources, and so these applications are not very polished. They're still relatively technical. They're not focused on members of the general public specifically. But it is our hope to make this data accessible to the general public. And that's why we made this data public. You know, we want high school students to be able to make sense of this data and really understand genomics not just by hearing about it through a press release.
Yeah, that's great. Okay. Another question. I might have to paraphrase a little bit. How much information is expected per genome? Once there are strong clusters and relatively few mutations, which are quite similar, let's see. I guess that's the main question. Like once you kind of know the population structure and I guess a lot of the mutations of the population, how much information is in a human genome?
So
I hope that answers this question for the.
So this is a great question, and I didn't get a chance to go into it when I talked about tiling and lightning, but our mentor George Church has been saying that the genomes are really not a big deal. They're only 4 megabytes. You can easily, a picture off your phone is bigger than 4 megabytes. So genomes are not big. Now, for me, a genome is hundreds of gigabytes of raw data that comes off a sequencer. So, how you get from hundreds of gigabytes to 4 megabytes seems hard. But the interesting thing with our tiling encoding is that we now have, in the preprint, we described an encoding that's about 40 megabytes. And we now have an experimental version of Lightning that can encode a genome in 4 megabytes, just as George had said. And so it is going to be possible to do real-time analysis on millions of genomes because a single genome can be represented very, very compactly. Now, the library, the library that genome has to refer to make that work, that library will still be gigabytes. It'll still be quite large. But it can be shared across many, many genomes.
Yeah. Great. Next question. Is it feasible to use AirVatos in clinical diagnostic setups like CAP, CLIA, accredited labs?
So the nice thing about open source infrastructure is that it is used all the time in CLIA and CAP accredited labs. And the way that works is that if you decide to use AirVatos, and people do use AirVatos in those settings, they go through a process, whether it's CAP or whether it's CLIA, in which they demonstrate that their process meets all of the CLIA and CAP guidelines. And so nothing about open source is an obstacle to that. If anything, it could make it even a little easier.
Yep, yep. While we're waiting for another couple of questions, I might ask, so you mentioned like Galaxy and other platforms. Could you compare AirVatos to those platforms, for instance, and how do you integrate or not with those platforms today?
Right. So we have a collaboration with the Galaxy project, which is really just getting started. It's around the common workflow language, which I mentioned. So essentially, when I talk about moving questions to the data, the idea is that you express them using the common workflow language. And the Galaxy project is thinking about using common workflow language as the way they express their workflows. And so it will mean that AirVatos and Galaxy can talk to each other through CWL. And so we hope over the next year or two to have this fully working in a way that allows you to just move back and forth.
Yeah. And so Galaxy, this is common workflow language, is something that is being industry adopted at this point?
Yeah, so common workflow language, about 80% of the spec was written by an engineer at CureVerse.
Okay.
And it has very broad adoption. Seven Bridges Genomics is a major leader of common workflow language. CureVerse is a leader. The Galaxy project is a leader. And there are many, many other organizations, several hundred bioinformaticians that are participating in implementing CWL.
Yeah. So let me just see if there's any other questions here for you. I guess still, so can you contrast, the model you showed is where you bring the computation to the data. Is Galaxy traditionally like a different model than that?
So Galaxy has both models. There are sort of centralized Galaxy instances that you can use as a researcher, and you can send your data there, and that's kind of like a centralized Galaxy instance. And then there are just local Galaxy instances that you can run near your data.
Right, right.
The big difference between the model that we're trying to implement with AirVatos and will be possible for Galaxy as well is to allow instances to talk to each other and to ask questions of each other's data.
Yeah.
So that's where things get actually very complicated because of security. So since your security permission framework and my security permission framework and all of the rules around those permissions could be quite different, that means that you need to do you need to have a lot more software infrastructure in place to implement that. And that's not something that's implemented in the Galaxy project today.
Yeah. No, I think, and the last question that I asked you, the last question that came in about the CLIA and the CAP certified, I think that actually is perfectly addressed by that, because their data could be private, as you mentioned, and
that's right.
And basically AirVatos can bring it to them, whereas other platforms they might have an option to play with right now, they don't have that option to use. That's great. Well, I think we're running out of time here, and so if there's no more questions, I think I'd like to wrap up the presentation. So I'd like to thank you again for the presentation, and have you any final comments for our viewers?
No, I think one of the things that is a little bit hard to wrap your head around with public data and open source software and these open standards is how do you fit in? You have private data and you have your own software that you're developing, and so I urge you to look at what's been happening in internet scale businesses where there's this collaboration between the open projects and your own thing that you're doing in your organization. And so there's absolutely a way for you to take advantage of these resources. And the place to start is to go to airvatos.org or to go to Github or to go to the URL that I showed for downloading this data and take advantage of it. It's there for you.
Great. All right, well, thank you once again, Sasha, and I'd like to thank our sponsor, LabRoots, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing after today's live event on the LabRoots website. You will receive an email from LabRoots letting you know when this is available for replay. Please share that announcement with your colleagues who may have missed today's live event. And so that's all for now. Thank you for joining us, and goodbye.